Then share my screen. Oops, that's not what I need. Get that out of the way. Okay, we finished up our first PowerPoint, and you're probably like, what on earth? That's not botany. Well, this is kind of basic biology. So let's go do some more traditional botany. So, plant of the day. Does anybody have an idea of what this plant is? Any ideas? Nothing? Have you ever heard of a corpse flower? Anybody ever heard of a corpse flower? Okay, there's actually two flowers, and I'll be showing you the other one at some point, that have the term corpse in the name. There may be more than that, but there's kind of two groups that I'm familiar with. This one is the corpse lily, versus the other one, which is maybe sometimes called the corpse flower. And let me get Sarah in here. So this is the corpse lily. The difference between the two, and I'll see if y'all can remember that when we get to the other one, is that this is a single flower. You can see that there are 5 major petals, whereas a corpse flower, the one people are more familiar with, I had some in the greenhouse, some small ones, but I don't think that they have survived. I need to see if I can get some more at some point. But they're not the biggest flower, they're the biggest inflorescence. So an inflorescence is a bunch of flowers together, and sometimes called a spike. In the case of that one, it's a spathe and a spadix. And both of them are known for being really, really large. And as you've probably guessed, the term corpse has something to do with it. What do you think is probably going to be the case for a flower that has corpse in its name? Why do you think it has that term? Let's use some critical thinking. Poisonous, that's a good guess. It's not the case, but that's a good guess. Let me guess. Were you thinking if you touch it or something, it'll turn you into a corpse? It's kind of the idea. Completely good thinking there. Okay. Elena says that it stinks, and that's actually the case. And guess what it smells like? It smells like rotting meat. So imagine you're walking through the forest and you smell this wonderful smell of rotting meat. You might actually think that it's some dead animal, but on occasion it's one of these flowers. And why do you think they smell so bad? Just bad hygiene? Defense mechanism? That's a really good idea. In this case, it's not at all a defense mechanism. It's almost the opposite. It's an attraction mechanism. So, are there insects that are attracted to rotting meat? Yeah, there are. And so, like beetles, flies, think about flies and maggots and all that stuff. Doesn't that make you like just want to go and smell this flower? So the pollinators are actually attracted to rotting meat. Now, this plant is also cool for a few other reasons. This corpse lily, as I said, is the largest flower of any flower in the world. It's like huge, 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 huge flower, a single flower. It smells like rotting meat, so it uses insects that like rotting meat for pollinators. And the other thing is this is actually a um, parasitic plant, and it grows on the roots, attached to the roots of other plants. Okay, and off the top of my head, I'm forgetting, I think it's a certain species, and I feel like it's in the grape family. So for the most of the year, and I think in some cases, multiple years, you never see it. It's actually completely underground, tapped into the roots of this other plant, getting its nutrients by parasitism. Okay, it's basically a little vampire, sucking the lifeblood out of, well, the life phloem um, out of this other plant. And the only reason we ever see it above ground is it uses the flower mechanism for reproduction. So to make more of itself. Okay. What do y'all think about that? Any thoughts? Would y'all be this quiet in the classroom too? Y'all are too quiet. Okay. If y'all are gonna be quiet, I guess I'll have to go lecture. Okay. Can y'all see the whole screen all the way to the side there? Okay. I'm trying to have my pictures of y'all up so I can see y'all in this as well. Thank you for the videos, and I understand that some of you can't do videos right now. As much as you can, let's do them though. And please feel free to jump in and say something. Botany is a whole lot more fun if we're interacting. Okay, plant cells. Cells were discovered in 1665 by Robert Hooke. In fact, they were plant cells that he found. The first cells discovered were discovered by Robert Hooke, and they were cork cells. You probably don't, think about cork board, but natural cork board actually comes from the cork cambium of the cork oak, which grows in Europe, Italy, and you strip this off the tree. It's a sustainable product, as long as global warming doesn't knock the trees out of existence. Now, why were plant cells the first cells ever discovered? Well, it was, one, the microscope was very, very rudimentary. And this probably didn't have a whole lot of magnification. And plant cells are, on average, the largest of all living cells in most cases. Okay, some of the largest cells in the world are actually from plant cells, so they're easier to see. Cork is also really easy to put under the microscope, and those cells were dead, so they were rather empty, and they're kind of squarish because of the cell wall that's remaining. So for him, when he looked at them, they looked like the cell, like a room. Okay, so hence they were called cells. And if we think about it, we use that word as a gel cell. Same idea. That's where that comes from. So these are thought of as little rooms, and that's where they get their name, even though they're not, rooms. It's kind of a strange thing at this point to think of it that way, since we know so much. But at the time, that's all it looked like. Cell theory later was developed around 1838 by Schleiden and Schwann. By the way, their stuff is really cool. Like, in my opinion, even though it's old, their development and understanding of how they understood what cell theory is really interesting. Now, are y'all familiar with the idea of cell theory? Have y'all heard that before? Does anybody know what cell theory is? Could you explain it to me? And I'm going to ask that somebody do so. If so for those of you that know it, or even just part of it, you can tell me part of cell theory. This is a very fundamental aspect of biology. Anybody? Anybody? Nothing? Does anybody even see the bullet point that's right below the cell theory bullet point? Like part of the answers on the screen. Okay, so living cells, living organisms are made of cells. That's part of it. That's one of the three major points. Living things are made of cells. Cells are the basis of the unit of living organisms, and cells make other cells. So for you to have life, you have to start with at least a cell. Okay. And so this gives us a very unifying structural basis of organization for living things. And it's cool because the first things that we see are plants. Now in Botany Lab, we will be looking at not microscopes themselves, but we will be looking at microscopes, but we'll be looking at slides under the microscope, and we'll be doing a lot of this, looking at cells and other structures made from cells. So we will have lab today, by the way. I'll see y'all. This is kind of what he would have seen under the microscope. Very, very simple. These are dead cells, so he didn't see anything inside them. They're just little squares or squarish, circle-ish, empty holes. So some of the first cells were, just basically a repeating little shape that someone saw. Just a little side note, cork is used for a lot of things. We're familiar with cork board, but it's also used for flooring. It is naturally antimicrobial, so some people will put this in their kitchen, and it's also very, I'm trying to think of a good word. I'm going to go with squishy because I can't think of a better word. So if you're spending a lot of time walking around on this kind of floor, it kind of gives, so it's better on your feet. Okay, think about those really nice mats that you might buy, put in front of the sink, because since you have to stand there and wash dishes for so long, you know how nice it is on your feet. Imagine a floor that does that to some degree, maybe not quite as squishy. So just a little side note. Okay, we'll be looking at microscopes. So light microscopes are what we'll be using. There are other kinds of microscopes. I'll briefly mention those. They increase the magnification as light passes through a lens. There may be a series of lenses made of glass or calcium fluoride crystals. You get compound microscopes, which we'll be using the most. And this can give us our largest magnification, up to 1,500 times. We will not be magnifying up to that much for plants. We usually only go about 1,000 times with bacteria, but it is possible to go to 1,500. You can actually get down to distinguishing organelles that are 2 micrometers or larger in diameter apart from one another. We will, in some degree, maybe sometimes be using dissecting microscopes, which can go up to 30x magnification. They're also called stereo microscopes. There are also field microscopes that you can take out with you to look at different things under the microscope. We will not be using these, but I will show some pictures that are from these. Electron microscopes use a beam of electrons that are produced with a high voltage electricity when you pass it through a wire. And there are 2 kinds of electron microscopes. One is transmission electron microscope, and they can produce magnification of up to 200,000 times. But for this to be done, it has to be sliced extremely thin. So if you're looking at a cell, you actually have to slice that cell in very thin little pieces to be able to see anything. And I may not show pictures of that, but you can find pictures of TEM um, specimens. Um, Scanning electron microscopes uh, go up to a magnification of only about 10,000 times, still quite a bit, but not as much as a TEM. But SEMs are still cool because they give you a lot of surface detail. So you can look at thick objects, the outer parts. And in plants, I'm more familiar with using SEM. If you are um, maybe working in geology as well, you might use SEM. It does get used in some other fields. So here's what a TEM might look like of a monocot pollen grain. So you can see that they've taken it and sliced all the way through, and you can actually see inside. And very cool, you can actually see little bits of the components of this pollen grain. Now down here, we've got an SEM of a dicot pollen grain, and this is the outer part. I actually really like the SEMs much better, more from an artistic standpoint, because I think these would be cool to look at. By the way, TEMs would be used more for, I think you might get viruses better because they're so small. I'm not sure if SEM will pick up a virus. I'm not sure about that, but just a little side note. The other cool thing about SEMs and pollen grains is SEMs are used to get these pictures of pollen grains, and you can identify the genus, not necessarily the species of a plant, based on the pattern on the outside of the pollen grain. This is a study called palynology. Very important for looking at historical ranges of different plants. The sediments at the bottom of lakes, sometimes caught in ice. If you can find pollen grains from sometime in the past, you can actually identify what was in that area, at least to the genus. You might not be able to identify it to the species, but close enough. You can also get a scanning tunneling microscope, and this is a probe that tunnels electrons under a sample, and you get like atomic resolution. We don't need that in this botany class, but just to let you know, they are there. So we'll be using these two pictures down below. On the left is a kind of compound microscope, and on the right here is a dissecting scope or a stereo microscope. We'll be using something like these. Ours look a little different than these, but they do the same thing. Now, review some stuff. Hopefully you know this. Prokaryotic versus eukaryotic. By the way, humans, what kind of cells do we have? Prokaryotic or eukaryotic?
Eukaryotic.
Yep, we are eukaryotic. I'm so glad that was a fast answer. So prokaryotes are cells lacking a nucleus. They are also lacking mitochondria or any other membrane-bound organelle. Eukaryotes have a nucleus and membrane-bound organelles, such as, well, you tell me, what are the membrane-bound organelles? Y'all should know this. What are the two major ones that we would talk about? There are others, though. Plants and animals. What do we got? There's one that we share and one that only plants have.
Mitochondria and chloroplast.
Mitochondria and chloroplasts are the two most common. Yep, there are others. We will talk a little bit about plastids more as we get along, briefly at least. Okay, plants have a cell wall. Are plants the only kingdom of organisms that have cell walls? Does anybody know of another kingdom that has a cell wall? Not a cell membrane, but a cell wall. I said kingdoms. You only have too many. You don't have any choices. Protista. Some of them probably do. Yes, I would not say all. because Protista is the catch-all. So let's eliminate Protista because that's true for some of them. What about another kingdom? So we've eliminated plants and Protista. How many does that leave you?
Fungi?
Fungi. Yeah, fungi have a cell wall of chitin. I'm trying to see, who did I have for bio 2? Catalina. Do I have anybody else in bio 2? Guess not. I know I talked about that in bio 2. I wondered if y'all had that in bio 2 as well. Now, plants have cell walls, but they don't produce the same kind of thing that fungi do. So fungi have something called chitin, and plants are made of cellulose. It's called a hemicellulose matrix. So you've got the plasma membrane that we might also call a cell membrane that surrounds the cytoplasm and that holds all the other cellular components, as well as the liquid cytosol. And then outside of that plasma membrane, you've got that cell wall. What does a cell wall do for plants?
Gives it structure.
Okay, it gives it structure. Okay, so it may keep it from expanding too much. It gives it a place, to kind of limit its shape and size. It gives it strength as well, and that will come into play later. So you can kind of see the boxy shape of a plant cell, which is based on the cell wall. The cell wall has space in between the two cell walls of two different plants. This is called the middle lamella, and you've got that space in between. There's even an intracellular space in between the two cells, where the kind of middle lamella gets expands, I guess you could call it. You can see things like chloroplasts, the mitochondria. Based on the picture, which one's bigger, chloroplast or mitochondria?
The chloroplast.
Chloroplast, and that is pretty normal, okay? So that's something to keep in mind. Chloroplast and mitochondria, don't consider them equal, okay? And size is a major component to understanding where those two things came from. So that's a good observation to keep in mind. Okay, eukaryotic cells are generally 5 to 100 microns. Cells of higher plants generally vary from 10 to 100 microns. So plants are on the higher end. Animal cells are on the lower end of size. And there are some plant cells that are actually larger than and longer than 100 microns. So just plant cells are huge compared to everything else. The cell wall is made up of cellulose, and it's made of a matrix of hemicellulose, pectin, and glycoproteins. The middle lamella is first produced when the new cell walls are formed. So you get that space in between. Secondary cell walls, so there's actually 2 layers of cell walls in some cells in plants. And secondary cell walls are derived from primary walls when you thicken them and you include lignin. Is anybody familiar with the term lignin? Has anybody heard that term before? You can just say no to help me. Okay, that's fine. I mean, if you just stare at me, I don't know if you've understood me or not. Okay, so I'll take some no's and yeses if I can't hear you. Okay, lignin, you may hear the term in things that deal with wood or paper. So we actually use wood products, and the reason we use it is because of lignin. And so basically secondary cell walls are what caused wood, and wood is what we use, and it's because of lignin that we can do that. So this is kind of a crisscross of so you can see the middle lamella, the primary cell wall, the plasma membrane. That primary cell wall, you can see how it gives it strength, where you've got this crisscrossing hemicellulose and these cellulose microfibrils. How many of y'all, I don't know if you make things, or like for example, my thought right now is going to a concrete wall. Has anybody ever seen a concrete wall made? And if you notice, they put rebar in it. You know what I'm talking about with a rebar? To me, that's kind of what this cellulose matrix is a bunch of crisscrossing rebar to give strength, and then they fill it up with the concrete around it. And that's kind of what we have going on here. We get that strengthening of these cellulose components, the hemicellulose and the cellulose microfibrils crossing and crisscrossing, and you strengthen things. Hopefully y'all have made enough stuff in your life, little crafts or whatever, that you learn if you want to build something, you want to build things where it crisscrosses to gain strength. I don't know if y'all have ever like tried to build a fort from sticks or anything like that. Have you ever done anything where you've had to crisscross something to give it strength? Can you give me some examples? I'd love to hear what y'all done, even if it was just something as a kid. Where have you seen this in play in your life outside of building concrete walls?
Lincoln logs.
Lincoln logs. Oh, great idea. Okay, yeah, you have to crisscross those to give them strength, and the more you crisscross them, the higher you can go, right? Has anybody ever played Jenga? Okay, what happens if you were to like line all the blocks up the same way? It's not one thing anymore. It's like just 3 lines of blocks, and it falls over, doesn't it? Please tell me y'all tried that. Anybody tried that? Y'all experiment as kids? Come on. Okay. It doesn't work. You have to actually get to keep crossing some blocks to be able to keep it, built up. Okay, same thing's happening here. Okay, now the cellular components, let's move inwards. So if you notice what we're doing, we're starting at the outer section and we're moving inward. Plasma, or sometimes called cell membrane. By the way, does anybody know why it's called plasma membrane and not just cell membrane? Why do we use the term plasma? Anybody know? I love science history. I didn't know for a really long time either. It's because some of the first cells that they extracted and tried to understand the membranes was from plasma in blood. So it was called the plasma membrane, the membrane of the plasma. So that's how it got that name. But it's kind of been expanded to cell membrane because we know that it's around all cells, not just plasma. Okay, we know that it's composed of phospholipids arranged in 2 layers with proteins interspersed throughout. Some proteins extend the entire width of your membrane, while others are embedded on the inner or outer surfaces. So I'm hoping y'all are familiar with the phospholipid bilayer concept. Do we need to go over that? Are y'all okay with the phospholipid bilayer concept? Okay, Kyra and Elena are. How about the rest of you? I have a question. Would you admit, kind of, okay, well, that kind of answers my question. I was gonna say, would you admit if you didn't know something? Kiviana's like, yep, I will. Tyler, would you admit it if you don't know something? Okay. Ikeria, would you admit it? Okay. Let's see. Brandy, would you admit it? Okay. The reason I ask that is because I don't want you to ever feel stupid when you're like, I don't remember that. Because I remember being that way as an undergrad and even before that. I'm like, oh no, I feel so stupid. I can't remember what they're talking about. Did I learn that? What class would that have been? I think all this stuff and I don't want to look stupid in front of other people. And so I never asked. And there's so many things that I didn't really get when I should have gotten them because I was too embarrassed to ask. And I just want y'all to know that you can feel free to ask. Um, it's worth that ability to learn at this moment and not have to go, okay, I don't know what he's talking about. And then you get to the test and you're like, oh, great, I really didn't understand that. So just feel free to ask me, okay? So phospholipid bilayer. So I've got a picture of it here. Um, so there is a head and two tails. So you'll see this ball that represents the head, and then there are two tails. Each one of these balls has two tails. Um, so there's a phospholipid, and those two, the name tells you a lot. So this is a head of phosphate, and there are two lipid tails. What do you know about lipids? Lipids are what? You have carbohydrates, lipids. Go for it, somebody.
Fats.
Okay, fats, oils. What happens when you put an oil with water?
It doesn't mix.
It doesn't mix. Okay, so what happens is we've got my fingers are the tails and the ends of my hands up here and here are the heads. So if they were moving around, what would happen would be that the cytosol is just made of a bunch of water, right? We're mostly about 70 to 75-ish percent water in our cells, and that's true for plants too. So you've got these phosphate ends that are water loving, and then you got these tails which are water fearing, hydrophobic versus hydrophilic. And so the hydrophilic outsides orient outwards towards where the water's at, and the hydrophobic sides orient towards each other. And we create this kind of layer of an area that's hard to cross. Okay, and that's what plasma membranes are, because inside the cell there's a bunch of water, right, in the cytosol, and outside the cell, guess what, there's also a bunch of water most of the time. Okay, now that's not true for our skin, but it is true for most of our cells because we have water-based components around our cells too. So what it does is it causes a barrier for things to cross into your cells or out of your cells, which is a good thing because you want the stuff to stay in the cell where it's supposed to be, right? And you want things that shouldn't get in the cell to stay outside. And that barrier of hydrophobic area makes it difficult for things to get through. Some things can get through. Water can go through, but more than likely, water and most other stuff go through little channels. These are the proteins. Okay, so like this protein here, you can see that there's a space for things to pass through. And I'm not going to get into active transport and passive transport. Y'all probably heard that in other places. Does that sound familiar? Okay, but that's where this is taking place. You've got to have things transported through the phospholipid bilayer. Now, also on the outside of cells and inside of cells, some of these proteins don't go all the way through, but they act as receptors for other proteins, for hormones, so basically for messages. So that can also be the case of things that are embedded within the membrane. Okay. Does that help everybody? Kind of remind you of what's going on? Okay. Does that sound familiar? Okay, gotcha. So communication between cells. We've kind of talked that you can get receptors in your plasma membranes, and you can, or hormones can be passed through and be received on those receptors. A lot of times fluids and dissolved substances can get through these membranes, like I said, on occasion through the hydrophobic, but more than likely it's going to go through something else. And in plants, there is something called a plasmodesmata, or desmotum, actually, in this case, a singular, but plasmodesmata. And this one's a little different than what we get with animal cells because we have something, plants have something that animals don't. So do you remember, we just talked about it. What component of the cell is the most obvious one that plants have that we don't? There's like two answers to this question. One of them's the one I want.
The chlorophyll.
Okay, so the chloroplasts, and in this case, that's not the one that I want, but that's true. So what's the other thing that plants have that we don't?
Cell wall.
Cell wall. Exactly. Thank you, Elaine. I see that too. Now, cell walls, do you think they could constitute a barrier between cells? Yeah, they can, just like a plasma membrane. And imagine if you've got a membrane and a cell wall, it's going to be that much more to try to get through. Elena, do you remember this from Plant Phys, the way that you had to track the different transports through the cells and around the cells? So one of the ways that plants can get through these things is to have little holes in your cell walls. And basically what plants do is, instead of having little separate cells that are abutting each other, we have little cells that are abutting each other that have holes that allow for little bits of cytoplasm to work in between. So you get little strands of cytoplasm. So do you see what I'm getting at? All of these cells are connected. Like they actually share parts of their liquid inside the cell in between these little plasmodesmata holes. Okay, so here's a TEM. You can see 1, 2, 3, 4 plasmodesmata. You can see a kind of like a colored version of this. So we've got the cell wall, the middle lamella that it crosses, and these little connections of cytoplasm. And then you can see a kind of blown up area of this. Why would it matter so much for plants to have plasmodesmata? What's the benefit of having these connections that are always there?
So stuff can get transported to each cell.
Well, couldn't they just work their way through the cell wall and the cell membrane? Why do you have to have connections? You're right, but why? Do I need to pull out the Jeopardy theme song? I can sing it. I really don't want to hear that, I'm sure. So what would be the use in connections? Okay, so let me ask you this. Which one's going to be faster? If you've already got a hole in the wall and stuff is traveling through, or having to cross a cell membrane, a cell wall, a middle lamellum, a cell wall, and another cell membrane? Which one's going to be faster?
The hole in the wall.
A hole in the wall, yeah. I don't know about y'all, but when I go from room to room, I much prefer to go through a door. It's a whole lot more work to actually create a hole in the wall to get through, don't you think? Same idea here. So this speeds up the process of communication between cells and sometimes passing and sharing nutrients and things like that. Okay. See, that was an easy answer, right? Y'all probably were thinking about those anyway, weren't you? How many of y'all think that you were kind of already on that line? Anybody kind of like already there? But just we're afraid? No, not for this one. Okay, that's fine. A lot of times my students, they're like, oh yeah, I know that. Okay, it's okay. This is probably the first time y'all have heard of a desmotubule. Okay, maybe not for Elena because like you had me for plant phys. We didn't talk about it much. So it's kind of a new thing. Okay, now the stuff that you are familiar with, and I'm not going to go into great detail because, hey, as animals, we have these, and you know, if you're going to take a cell biology class, you know, like how many of y'all have had cell and molecular? Somebody had cell and molecular? Okay, you've had cell and molecular. Okay, some of y'all will be having it probably next semester. A number of you probably. So you're not going to get into great detail here because you're going to get this in other other classes. So plants have nuclei, just like we do. They're eukaryotic organisms. They have endoplasmic reticulum, and endoplasmic reticulum sometimes is abbreviated as ER. So there is smooth ER and rough ER. Can anybody tell me what the endoplasmic reticulum does? Or at least just like something about it. We need to work on that. So bio majors, we got to know this stuff. A lot of times, not all of it is just this, but one of the difference you get with smooth ER and rough ER is ribosomes. Why is the ER smooth? Well, it doesn't have ribosomes on it, and the sections that are rough have ribosomes on them. Ribosomes are for protein construction. They're used in translation of the mRNA that comes out of the nucleus. And so you've got protein construction and packaging in the endoplasmic reticulum and sometimes throughout the cytoplasm. And in plants, we have another thing called the dictosome, which you probably haven't heard of, but basically it's just the Golgi apparatus or Golgi body that we might call it in animals. Just sometimes you'll see that, especially in older textbooks, the plant names for things are a little different than the animal versions. But as we move forward, a lot of plant stuff is getting animal, basically starting to use animal names. So what does the Golgi do? The Golgi is about packaging those proteins a lot of times for export outside of the cell. Those are basic ideas at least. So you probably want to remind yourself of these things at some point. Now, plant-specific stuff, chloroplasts, okay? These are the most conspicuous plastids. This is a group of things. Each one is bound by a double membrane. They contain a stroma, an enzyme-filled matrix. They have DNA, RNA, and ribosomes inside of them. They contain grana, which are basically little stacks of thylakoids, and thylakoid membranes have chlorophyll in them inside of these chloroplasts, and it increases their surface area. Surface area is one of those things in biology that's so important. If you understand how surface area works, you're going to see a lot of things making much more sense as a as a biological process. So why have large surface area? So you can capture more light. Well, how do you do that? Well, there's a lot more chlorophyll in that thylakoid, and the more thylakoid surface area you've got, the more chlorophyll you've got, the more light that can be absorbed, and the more light you can have absorbed, you can do the first parts of photosynthesis, which will then allow you to do more in the stroma and create sugars, and then bam, you've got more food. Okay, so the more surface area, the more food in this case. So yeah, photosynthesis, that's what it's about, chloroplasts. Okay, let me see. I'll come back to that in a second. I do need to ask you about the double membrane. Why do chloroplasts have a double membrane? This goes back to the origin of chloroplasts. And also, mitochondria. So I'm trying to ask, like, why do you think chloroplasts have two membranes instead of one? Yeah, endosymbiosis. So endosymbiotic theory tells us that more than likely chloroplasts were absorbed as probably single membraned, maybe cyanobacteria, and instead of being destroyed or consumed, they ended up being incorporated into the plant, and at some point the DNA inside of them was somewhat shared with the nucleus of the plant cell, and you get a symbiosis between the two where you get them the ability to make their own food, and plants become plants this way. Okay, the eukaryotic cell absorbing a cyanobacterium, which becomes a chloroplast, and it gets a double membrane because as it was absorbed, probably the outer membrane of the cell enveloped it. So you ended up with an extra membrane from the plant cell there. Now I'm going to stop with this slide. Just and here's a picture of chloroplasts actually moving around inside. If everything goes well, we'll hopefully be able to see moving chloroplasts under the microscope in lab. Not today, but some days. Okay, someday in the future. Okay. Also, a couple of things for lab. Today, I'm going to bring the lab handout for you. But after today, I'm going to ask y'all to bring it yourself, okay? But I'm going to print them out before lab and bring them up to you, okay? We're going to meet in room 230 at 2 o'clock. And yeah. Sarah, are you going to be able to join us? Do you know? Yes, I don't know where she's at. Okay. Okay. Let's see. Well, that's all I've got for right now. I'm going to stop the recording